Hi everybody, we're back at it again with a new video. Today we're going to be going over DNA sequences and the different mutations that we can observe. Here we are given two different DNA strands, our template strand and our coding strand. The process of changing DNA to mRNA is called transcription, and we are going to use the coding strand to achieve that. We can think of mRNA and DNA as a pair of shoes. DNA uses thymine, which is the left shoe, and mRNA uses uracil, which is the right shoe. Both shoes look the same, but we can't put a left shoe on a right foot and vice versa because it just won't fit right. The right shoe, uracil, just does not fit with DNA, and the left shoe, thymine, just doesn't fit with mRNA, despite the fact that they look the same. To make sure we get the proper mRNA strand, we change all of our T's to U's so that it now fits correctly. In this example, our coding strand is written 3' to 5', left to right, but we want it in the 5 to 3'. So all we have to do is flip the strand, making sure to keep the order the exact same in relation to the 5 and 3' ends.
After everything has been transcribed, we have the translation process, which is the process that identifies the different amino acids in the newly transcribed mRNA sequence. Here are the amino acids observed. We only code for the amino acids that occur once the start codon, methionine, has been observed, and stop coding when we see one of the multiple stop codons, in this case UAA. The rest of the sequence is no longer something we are going to look at.
In these strands, we can observe point mutations, which are mutations in one single nucleotide in the different DNA or mRNA strands. There are missense, nonsense, silent, and read-through point mutations, and I'm going to give an example of each one and show how it affects each new strand. We can also label the mutation transition, which is changing purine to purine or pyrimidine to pyrimidine, or transversion, which can change from purine to pyrimidine or vice versa. Using another shoe analogy, we can think of a transition mutation as changing from one pair of sneakers to another pair of sneakers, which are similar shoes. And we can think of a transversion mutation as changing from hiking boots to high heels, which are two different styles. First, we have our missense mutation. This mutation simply codes for a different amino acid than the original strand. Here you can see that the new strand changed from AGG, which codes for arginine, to AAG, which codes for lysine. This transition mutation because we changed purine G to purine A. Next, we have our nonsense mutation. This nonsense mutation changes an amino acid into a stop codon. The sequence changed the AAG lysine sequence to UAG, which is a stop codon. This is a transversion mutation because a purine A changed to a pyrimidine U. Our third example is our silent point mutation, which means that the new sequence still codes for the same amino acid. The sequence shown changed from GUC valine to GUG, which also codes for valine. This sequence was also a transversion mutation because the pyrimidine G changed to purine C. Finally, we have our read-through point mutation, which is a mutation that changes a stop codon to an amino acid. The sequence shown changes stop codon UAA to CAA, which codes for glutamine. This is a transition mutation because pyrimidine U changes to pyrimidine C. Thank you guys for watching.